Okay, today we're going to talk about cancer immunology and immunotherapy. This is an area which initially looked very scary to me. We started working on cancer immunology about 5 years ago, but now that we have learned some of that, it's actually really, really fun, and I hope you all enjoy this. Can you help me close the door so it's not too loud? Thank you. So if you remember the earlier lectures about different mutations in different cancer types, there was one common understanding that there are more mutations for tumors in older patients and tumors facing outside. For example, melanoma, where the patient, over time accumulate mutations from radiation, or lung cancer, even though it's inside the lung is really getting the air from outside, smoking, pollution, or all your GI tract, whatever food you eat, the bacteria in the gut or alcohol you drink. These are all things, basically facing outside. And so a lot of these tumors have more mutations in the tumors, and they are usually developed in patients who are a bit older. Whereas those that are developed in younger patients are like AML, leukemia, or brain cancer. Those can happen pretty early, and it's usually caused by one mutation, which caused something fairly acute. And so these tumors, besides that driver mutation, usually have very few other mutations. So even though those early tumors, like when pediatric patients, in early days, they are the most deadly cancers. Once you identify the driver gene, people can also develop a drug, and they are usually easier to cure. In the tumors that have, many mutations, even if you can identify a targeted therapy, sometimes these are really hard to cure. It's like, when you have a traffic jam like this, just a single policeman is going to have a very difficult time clearing up this mess. And so when you have a tumor with many mutations, if you just have one targeted therapy to cure, target one gene, because these tumors that appear in older patients with a lot of mutations, they usually have DNA instability. When you have a targeted drug against one mutation, the DNA instability can easily evolve to have additional mutations that drive the cancer, or evolve to have a mutation which prevents the drug from even binding to the target that we are interested in. And so these tumors that have a lot of mutations are usually harder to treat, and they are kind of more deadly. Things change in the last decade or so. So people are interested in immunology for a long time. Immunotherapy has also been under development, but in the early days, it was not very promising until about 5-10 years ago, that immunotherapy really started to make a difference. And in 2013, so every year, the Science magazine at the year end will rank the 10 most significant findings, breakthrough of the year. And in 2013, the #1 breakthrough of the year is cancer immunotherapy. And we know that last year, the Nobel Prize in Physiology or Medicine was awarded to James Allison and Tasuku Honjo for their pioneering work in developing immunotherapy. And within our lab, we started working on cancer immunotherapy in 2014. And in the very early days, we're just trying to learn the very basic, what is immunology? What are the important players? How does immunotherapy work? And so I want to also kind of give you some very basic introduction as well. So you ask, this unfortunately has been done before. If you take somebody's tumor and try to put it in some other people, would that grow in another person? Yeah, so people have observed that this has been done in mice, except that in the mice, these are usually immune deficient mice. The mice, you would engineer the recipient to not have either T cells or B cells, like immune deficient. If you just take somebody's tumor and try to put it in another person, you will have rejection right away. And so it means that our immune system can recognize this as this external tumor as not being his or her own, except occasionally it can grow. And in the early days, I think some mistakes were made, but people don't do this anymore. So in fact, so transform the cells just mean there are mutations happening, the cell become cancerous, right? Very often when this happens, within our body, we very quickly recognize that these are bad and it get eliminated. So our immune system really is helping to protect ourselves against not only viral or bacterial infection, but also against any cancer occurrence or mutations within our normal cells. And so, in fact, within all of our body, as the cells are dividing, error rate happens. It's 10 to the minus 9 roughly. So anytime there could be some mutations happening, occasionally you could have a mutation which makes the cell grow a little faster, or they do not die, or things like that. But very often our body will be able to recognize that. So young people, when you have more cell cycles, you are actually likely to have more cancer cells as well, except that our immune system can easily recognize them and wipe them out. For some reason, maybe our immune system get a little weaker, some of these cells will be allowed to stay on, and there will be kind of an equilibrium. The immune cells is trying to do some fighting or killing, but it's not quite killing all the cells, and so they are happening at the same time. These are called equilibrium. In fact, I think a lot of our individuals within our body, we have those little equilibrium spots, right? That nowadays, when people look at the mutations that happen in different tumors, they can see that some of these mutations happened like 30 years before the tumor is detected, which means 20 years before it's in this equilibrium state. And by the time we do diagnosis, it's usually when your immune system has failed. For some reason, there is an immune escape. And so the person, basically, the immune system is no longer able to suppress the cancer cells from growing. And so now the tumor is growing big, and that's when you detect them. And so in the very early days for immunotherapy, the simple thing is to just try to boost the patient's overall immune system. So here we introduce the term cytokines. So these are cell-secreted proteins. When encountering foreign particles, can stimulate cell growth, inflammation, differentiation, repair, and just various immune responses. Although I would say there are cytokines which activate some cells but repress other cells, because our immune system is very complicated. Some has activation function, some has suppression, like pro-inflammation versus anti-inflammation. Depending on the specific cytokine, they will have effect on different cells in very different ways. And so one of the earliest active immunotherapy is this cytokine, which is called IL-2. Yeah, this is FDA approved for kidney cancer and melanoma. Basically, you just inject this protein into the body, and the cytokine will stimulate it will have mostly immune activation function, stimulate all your immune cells. And yeah, so it boosts the overall immune inside the patient body. So you can see with IL-2, which if it's normally secreted, can increase the function of B cells, T cells, natural killer cells, monocyte, and T helper cells. So you have kind of an overall call to duty for a lot of our different immune cells. This you can imagine, yes, it helps the patient, but also it can be very toxic. Basically a patient will have fever, potentially autoimmune problems. Okay. What really made immunotherapy a breakthrough in science and cancer care is using immune checkpoint inhibitors. And that's what got James Allison and Tsuku Kanjo the Nobel Prize, is this immune checkpoint inhibitor. And so this is how things work roughly. So you can imagine the T cells and B cells in our body are like guards. They can have a receptor on the cell surface which recognize self versus non-self. So these receptors are matured so T cells mature in our thymus and B cells mature in our big bone marrow. And when they are maturing, the thymus and bone marrow has a way to show the cell all sorts of proteins that our body make. Say, okay, make sure this is ourselves, this is ourselves. So anything that recognize ourselves are eliminated in the thymus and bone marrow and not allowed to come out. And so those T cells or B cells that doesn't recognize ourselves are allowed to go into circulation. So they are kind of always patrolling our body to look for virus, bacteria, or cancer mutations, okay? So all of our normal cells has a mechanism called major histocompatibility, which is a protein complex, is a receptor on the cell surface, which routinely present degraded proteins onto the cell surface to allow our T cells and B cells to examine this to make sure that everything is okay. And so you can imagine, because all the things in circulation do not have a strong binding against anything to our cells, because if they do, they will be dead in our thymus and bone marrow. Anything in circulation is supposed to recognize other things. So all of our normal proteins being presented onto the cell surface, when our normal T cells, B cells see them, will say, okay, fine, these are pretty good. They don't have very strong binding. However, if you have a viral infection or bacterial infection, they will have some unusual proteins. Or if you have a cancer cell that has a mutation, this mutated protein can also be presented onto the cell surface. They will look very foreign. Therefore, there might be some T cells and B cells which see this right away and say, this guy is bad. I've never seen this. And there would be some T cells and B cells have very strong binding to this. The receptor will have strong binding to this. And in most cases, they will be able to cause different immune effect to eliminate the cancer cell. However, as we get older and older, our immune system get weaker. And also, by the way, for most people, your thymus function is the best around the puberty. And after that, the thymus function pretty much disappears. It's not working much anymore. And so the type of different T cells in our body that's in circulation is whatever was there. And if there is immune stimulation from recognizing some flu or virus or like a bacteria, it will proliferate, make more clones of it. Otherwise, it will die. And so if you look at the different T cells, what's the amount, diversity of the different T cells as we age, the T cell diversity decrease over time from puberty. And in fact, male decrease faster than female, maybe because of drinking or fighting or working too hard or whatever reason, staying up too late, playing video games or whatever, they decrease faster. And this is probably why male have developed cancer more frequently than female. But in addition to having, so you can imagine if that particular mutation, which is supposed to be recognized by a particular T cell, and that T cell died over time because it never gets a chance to be stimulated, it will die. And next time this mutation happens, then it will be allowed to grow because the T cell that recognizes it is gone, right? So occasionally that could happen, but also, so that's when this equilibrium might happen. But also the cancer cells sometimes over time can evolve mechanisms. For example, in this case, they secrete different proteins like ligands, which can interact with receptors on the T cells. You can imagine this like, alcohol passing to the T cells to say, hey, buddy, we're not too foreign, we're just friends, stay calm. And so these ligands, when they interact with the receptors, will make the T cells or suppress the activation of the T cells or make them something called dysfunctional. So they're no longer getting activated. Very often, if you look at a tissue slice for a tumor, the T cells are right next to the cancer cells. They should be recognizing the mutation killing, but they are not, right? So very often it's because the cancer cell, the T cells get drunk from the ligand secreted by the cancer cells. And so with this type of scientific discovery, then the immunologists developed therapeutic antibodies. They either target, in this case, PD-1, or PD-L1 is the ligand, PD-1 is the antibody. And there's also anti-CTLA-4, which basically can bind to PD-1, PD-L1, or CTLA-4 stronger than the other counterpart. And so they block the ligand from interacting with the receptor. And this effectively is like pouring ice water onto the drunk guards. And they will just, wake up, right? So once they wake up, they are back to, working stage. And very often, they will recognize this as, this guy looks totally different, right? Or they'll have strong binding and get activated, and that will kind of eliminate the cancer. And so this is really very cool because you can imagine in the early days when people were developing immunotherapy, they are trying to use a protein, say the PD-1 antibody, which is really, I think the most exciting development is you're developing an antibody against your immune cells. And when that works, this immune cell is activated to kill the cancer cell, which is really, really cool. It's not directly killing the cancer cells anymore, right? So it's very, very promising, except that only minority of the patients in very small number of cancers currently this has been FDA approved. Yes, Jet.
Do the patients that either don't respond or do respond, do they have issues with like autoimmune problems?
So the question is whether patients who don't respond have autoimmune problems. Most of the case, patients who have autoimmune side effects are more likely to be those who responded. They just had a little stronger immune response that they can't tolerate anymore. Whereas a lot of patients who couldn't respond. Well, I guess there are cases when the side effect from the autoimmune is so strong, you could kill a patient from something called a cytokine storm. This person suddenly have such a strong effect, they die. But nowadays, people can manage this a little better. But there are various reasons why immunotherapy do not work, and we can show you a little bit later why these are the cases. Okay. For cytokine storm, it's kind of interesting. In the very early days, when scientists were developing immunotherapy, the first thing is they tried on mice. And the CTLA-4 antibody added, they probably didn't know how much to add. All the mice die right away because of very strong immune boost. So they have to control the dose. Okay. So besides the immune checkpoint inhibitor, which are antibodies to block the ligand receptor interaction, there are also CAR Ts, which have also been FDA approved. So the idea is like this. If you have a cancer patient, you take out you just get their blood, isolate the T cells, and you use some way to engineer this T cell so that the cell surface has a receptor. It's called a chimeric antigen receptor. So on the cell surface, it could be either a receptor or an antibody. And then once it binds to the correct thing, underneath, you'll have an activation mechanism to activate the T cells. That's why it's chimeric, because the top and bottom are engineered to do different things, right? So if the receptor recognizes the tumor antigen, then the downstream effect is the T cell can get activated and proliferate, which can then, does the killing. And with this engineered T cell, you can go in the petri dish to amplify 10 to the 9 or 10 to the 10th number of these T cells. Then you infuse them. It's like a special force, right? Like a Marines or whatever, Navy SEAL. You can amplify a huge army of them outside the body by adding IL-2. right? So IL-2 will stimulate T cell, they proliferate a lot, and then once you make so many T cells, you infuse them back to the patient. And because each of these T cells has a little receptor to recognize a tumor antigen, they will go all over the body, and every time they see the bad guy, they will go and kill, right? So that's a CAR T. And so this works very well, mostly very well in blood cancer, leukemia, lymphoma, because it's very well known what kind of receptors can uniquely recognize a cell surface protein on the leukemia lymphoma cells. So these are either CD19 or CD20, and you can have an antibody against them and engineer them on the T cells. They will work very well. It's pretty good, except that usually before you do the infusion, the patient needs to go through a chemotherapy to wipe out their kind of T cells or B cells to, in some sense, you know, sometimes before you plant a new field of crop, you want to scorch the earth. So you get rid of all the weeds and other stuff, right? And then the field is retilled, and then you seed all your new things to grow. And so usually the patient goes through a lymphodepletion to kind of wipe out their T cells and B cells temporarily. It's still in the bone marrow, but in the blood, you wipe them out. And then when the infused T cells are there, they are really ready to take in the seed. And so this works well, hopefully, but it costs about $600,000 for one shot of 10 to the 10 T cells. If it doesn't work, you have to try something else. Yeah, so this is like just CD19, CD20 on leukemic or lymphoma. For solid tumor, people are trying something called adoptive T cell transfer. So for example, this is in melanoma. So patient tumor, you can use a surgery to take it out. You isolate the cancer cell and the immune cells. And the immune cell, you put them into single clone to kind of give it IL-2 to let it grow up. And then in the cancer cell, you can just crush it up. And then each of them, you can put them in these immune cell little clones and see whether the immune cell can recognize the mutation on the cancer cell and proliferate. Because if within your tumor, there might be different type of T cells. They recognize different things. You want to amplify the clone that really recognize your cancer mutation. So you can have little tumor debris to a little bit in the different clones to see which one activate. And for those reactive clones, then you amplify a lot, right? So in this case, we don't engineer a receptor onto the T cells. It's whatever T cell that has the right receptor to recognize cancer get proliferated to 10 to the 10th again, then you infuse them back to the patient. Okay. This has also been tried. These are mostly research trials. There hasn't been a commercial trial like this, FDA approved. But can you imagine what type of potential problems are when the T cells in your body, infuse the T cells going to the patient body when they approach the cancer? Can you see what happens there? Jet.
I mean, I'm not sure if this answers your question, but one problem is that the biopsy itself may not be representative of the entire tumor. I
see. So one question is whether that particular tumor biopsy is representative of all the tumors in your body. I guess there are some early mutations that later on get propagated, right? So then if your T cells recognize those early ones, they will probably work for all. But one potential problem is the fact that this patient has tumor means they already have an immune escape, and so it means their cancer cells can probably secrete those ligands to make the T cells dysfunctional. And so you can imagine as the T cells are approaching the cancer, they get drink, they get really happy, and they can become dysfunctional. right? Something like that. So this can potentially be combined with immune checkpoint inhibitors, right? So not only you train troops outside, you arm the troops with the right things so that they go in and stay active, right? Something like that. So this can be combined. And recently, for the same group, this is Steve Rosenberg's group at NCI, they do personalized T-cell immunotherapy, which is mutation-based. So the idea is, you still get the patient tumor, you isolate the immune cells, you also isolate the cancer cells, and the cancer cells can go to whole exome sequencing for mutation analysis. And based on the mutations, they can predict which of those mutations are likely to generate an immune response. We will teach you this part a little later. And then they use dendritic cells to express, they use other cells to express this small collection of peptides which has the mutation. So supposedly this patient might have, 50 different mutations that are likely to be immunogenic. They can make these dendritic cells to express this peptide, which each of them is about 10 peptide long, and the middle point is that amino acid mutation which is observed in that patient tumor. So in some sense, you are highly expressing these mutations, and then you co-culture those dendritic cells in with the T cells, and any T cell that can recognize this will automatically amplify again, right? And so in some sense, it's like each of these cancer cells, instead of just getting a tumor fragment, you actually use dendritic cell to present this, highly express this mutation to be given to the cancer cell. To be given to the immune cells for recognition and proliferation. And then you amplify these immune cells again to like 10 to the 10th and inject them back to the patient. And that also seemed to have worked well. So interestingly, I remember in this Nature Medicine paper, they mentioned that, depending on the patient, the little peptide that they give to that patient's immune cells can be very different from the peptide they give a different patient, because their mutations are very, very different. And in this case, the majority of cases, they do see very good mutation-specific responses for the patient. Okay, so that's another way. So these types, their cost is going to be probably similarly high, like a CAR T, because you have to take somebody's tumor, also take somebody's immune cells. So most of the time in this case, they don't take the regular T cells from the blood. They just take the T cells from the tumor, because they are hoping that those T cells in the tumor were already there to recognize the mutation. They were just somehow getting dysfunction or getting drunk. And so they have the best chance to be reactivated outside the body. If you just stimulate it with the dendritic cell and also IL-2, they might kind of wake up again, right? And then you train them outside and then you infuse them back to the body. But growing 10 to the 10th number of T cells outside the body and make sure it's kind of clean enough you can inject back to the patient is not a cheap thing to do. I would imagine it will be something similar order, like $500,000 or so. So it's still an expensive experiment. Yes, Jet?
Does immunotherapy work for cancers that are derived from immune cells?
So cancers that are derived from immune cells are the liquid blood-related tumors like T-cell lymphoma, leukemia. Those CAR T works for like CD19, CD20. However, these immune-related or immune cell-related cancer usually happen in younger patients. And younger patients in general, like leukemia, lymphoma, you can use chemotherapy or bone marrow transplant. So they do have some other options as well. Okay. So for melanoma, it's usually later patients, which usually by this time is chemotherapy resistant. It's like very difficult to treat. And that's when you try these other immune-related things. Next one is cancer vaccine. These are, most of the time when you do a flu vaccine, it's preventative, prophylactic. But cancer vaccines are mostly therapeutic. You do not, so far people haven't really done a vaccine which can prevent cancer, except for, viral infection. Like if you get rid of HPV, like those, yeah, definitely you reduce liver cancer, gastric cancer. Those are different. But in this case, the most of these cancer vaccines are when the patient has already been diagnosed with the cancer, and then they try to do something. So in this case, you take out, say the dendritic cell. So I will talk about this a little later. Dendritic, so in your body, we have different way of presenting the antigen. Your regular normal cell can present all the internal peptide to the outside for examination. This is you need every cell has that mechanism, but there are also something called professional antigen presenting cells. So these things, basically, when they see bad things, they eat it up and chew it out and then present it on the cell surface. The dendritic cell is one of those. And so you can take the dendritic cell, and there you can use the tumor lysate. You can also use a viral vector or some peptides which are kind of cancer mutations, and you kind of train your dendritic cell. You can make the dendritic cell just express these things, or you can use your tumor stuff to also recognize them, and then you can, in some sense, bring this into the body. People also recently try to use these viral vectors. It's called oncolytic virus. So interestingly, these viruses can just bring some mutations, or the thing is, once they infect the body, they usually go to the cells that grow fast, which very often is the cancer cells anyways. And because the viral thing has something that's not natural, it's like other foreign things. Once they go to the cancer cell, they present additional bad guys onto the cell surface, which in turn can activate your immune cells. And also when the virus is kind of circulating and trying to go to the cancer cells, it can also boost your body's normal immune system. It's like a small IL-2 to simulate the immune response. And so there are cases, for example, when a patient initially do not respond to a chemotherapy, but after they have a flu, they do respond to chemotherapy now. Potentially that viral infection did a kind of your immune boost. And some of that might have gone to your cancer cells. Yeah, so that's something that people have tried. But most exciting thing recently is personalized, a new antigen vaccine. So you can imagine if a patient has a tumor, if we can excise some of the tumors for, again, whole exome sequencing, based on the peptides or the mutations that are based on the sequencing, there are also algorithms to predict which of those mutations are likely to be immunogenic. And there, instead of training the troop outside, like training a guard outside, new antigen vaccine, basically you just make a tube of peptides or RNA which contain the mutated thing. So for the peptide, it's usually just 30 amino acid long, but you put in, say, 15 or 30 those peptides that are predicted to be immunogenic, and you inject the patient. In some sense, these peptides, when they are in the body, is training your body's T cells to say, take a look at these bad guys in some sense, right? And then the T cells will get trained, and then they will circulate and go and amplify in your body and go look for the bad guys. And this is cheaper than proliferating 10 to the 10th number of T cells outside the body. But it depends, I guess, on whether the patient in the blood, they have enough good potential T cells to be activated. And this can also potentially be combined with the immune checkpoint inhibitors, because the T cells can be trained, and as they are approaching the tumor, can also get drunk. And so you can use checkpoint inhibitors to wake them up. Okay, so these are all the recent exciting developments in cancer immunotherapy. Questions? Yes, Sam.
For childhood diseases that have specific oncogenes that get mutated, you use something like this as like an actual vaccine at like cancer. You know exactly what
So far, these new antigen vaccine trials has mostly been done in melanoma, and I think recently was another adult later stage tumors. We haven't seen this to be used in pediatric cancers with very, very few mutations. Okay, I can tell you why this is the case a little bit later. So